Welcome to this high-yield explainer on intestinal Taeniasis. If you're getting ready for your exams, you are in exactly the right place. We're going to break down everything you need to know about beef and pork tapeworms in a way that's super clear, quick, and easy to remember.
All right, let's just dive right in with a classic question you'll see in your practical exams. You're looking at the heads, the scoliosis of Taenia Solium and Taenia Saginata. How in the world do you tell them apart? Well, the key is that one of them comes armed for battle, and the other one doesn't.
And this slide shows that difference perfectly. Just look at image B. That's your Taenia Solium, the armed pork tapeworm. See that beak-like thing, the rostellum? It's got 2 rows of hooklets. Now, compare that to image A, which is the totally smooth, unarmed head of Taenia Saginata. You gotta memorize this visual. It's a classic spotter.
So here's our game plan for today. We're going to cover all the bases. We'll start with the basics, trace that wildlife cycle, check out the symptoms, decode the lab diagnosis, cover treatment, and then wrap up with that all-important high-yield comparison you absolutely need for your exams.
Okay, first things first. Let's start with the absolute fundamentals of intestinal Taeniasis. What exactly are we dealing with here? In a nutshell, Taeniasis is an infection of your gut. It's caused by the adult form of these 2 major tapeworms. And remember, humans are the definitive host, which just means the adult worm loves to live inside our intestines. Now, this right here is a critical distinction. You absolutely have to remember this. While both of these worms cause intestinal taeniosis, only Taenia Solium can cause a second, much more dangerous condition called cysticercosis. That's where the larvae actually invade tissues like the brain and muscles. So yeah, it's a true double threat.
Okay, so next up, let's trace the parasite's pretty fascinating and, a little bit unsettling life cycle. Understanding this journey is really the key to understanding how we can prevent it. This diagram maps out the entire journey beautifully. Let's start up at the top with us, humans, the definitive hosts. This is where the adult worm lives. If you follow the arrows, you can see how the eggs get passed into the environment. And then they're eaten by cattle or pigs, the intermediate hosts. That's where the larvae develop in the muscle. Let's break that down step by step. So to break it down, the cycle kicks off when a person eats undercooked meat that's got these larval cysts in it. Inside our gut, that larva grows up into a massive adult worm. This worm then produces thousands and thousands of eggs, which get passed in the feces. And if this contaminates the environment, cattle or pigs might ingest those eggs. The eggs then hatch, they migrate to the muscles, and they form cysts, just waiting to be eaten all over again.
So how does this infection actually show up in a patient? What are the red flags we should be on the lookout for?
You might expect a giant tapeworm to cause some pretty dramatic symptoms, right? But what's surprising is that most infections are actually asymptomatic. A lot of the time, the only clue is when a patient discovers these motile, ribbon-like segments, the proglottids, in their stool. When symptoms do show up, they're usually pretty mild and nonspecific, things like abdominal pain or a little nausea.
Okay, now for the really crucial part for your exams. How do we actually confirm a diagnosis in the lab? This is where the details really matter.
So here's a major diagnostic challenge, and it's a favorite exam question. Under a microscope, the eggs of T. solium and T. saginata are morphologically indistinguishable. You just can't tell them apart by looking. So, how do we differentiate them?
Well, the key is a special staining property. If we use a modified Zeal-Nielsen stain, the eggs of Taenia saginata show up as acid-fast, but the T. solium eggs don't. It's a really simple but kinda elegant way to tell them apart.
And here's a really simple trick to lock this in for good. Try spelling Saginata backwards. You get a Taeniasis. Sounds kind of like gassed, right? Now just change that G to an F and you get fast. Saginata is acid-fast. That's an easy point to remember on exam day.
Now, staining isn't our only tool in the box. The diagnostic toolkit also includes your standard stool examination for eggs and proglottids, anal swabs, which can actually be more effective, and of course, modern techniques like ELISA to detect antigens in the stool. But the gold standard for telling the species apart is going to be PCR, which targets their unique DNA.
Okay, so we've moved on to how we treat this infection, but more importantly, how can we prevent it from even happening in the first place? Prevention is actually beautifully straightforward. All you have to do is remember the mnemonic CD. C is for cook your meat thoroughly to kill any larvae, and D is for dispose of human waste safely to break that transmission cycle to animals. It's a simple public health message, but it is incredibly effective.
And here's the good news. Intestinal Taeniasis is very treatable. The drug of choice is a single dose of praziquantel, which is highly effective at killing the adult worm. Nicotinamide is another option, but really, praziquantel is the go-to.
And now for the climax of this whole explainer, the ultimate comparison chart. You're going to want to screenshot this, write it down, commit it to memory, because this is pure exam gold.
But before we even dive into the table, here's a super simple trick to remember a lot of the key differences. Saginata is a longer name than Solium, right? Well, this corresponds to it being a bigger worm, with more segments and more uterine branches. Bigger name, bigger features. Simple as that.
And this table lays out exactly how that bigger name, bigger features rule works. See? Saginata is longer, it has more uterine branches, and it comes from the larger host, cattle. This chart summarizes everything we've covered, from the Scolex morphology all the way to that crucial acid-fast stain. Seriously, this is your key to success on this topic.
Okay, let's do a super quick rapid-fire revision. For Solium, just think S. It's the smaller, Scolex-armed worm that causes serious cysticercosis. For Saginata, remember it's the larger beef tapeworm, and its eggs are acid-fast. Know the hosts, know the transmission, the main diagnostic points, and that simple one-drug treatment. You've got this.
All right, we'll end with a little clinical thought question to tie it all together. A patient tells you they ate undercooked pork. You find Taenia eggs in their stool. You know it's T. solium. So why do you have to consider not only intestinal Taeniasis, but also that much more dangerous systemic disease, cysticercosis? Think back to the life cycle and remember the unique double-threat nature of this parasite.